So I get the opportunity to talk about advances in analytical techniques, specifically near infrared, and really how we're taking that technology onto the farm. And the NIR story really starts in the 1800s with William Herschel, who discovered there was this strange energy at the end of the visible spectrum which caused heat to be generated. And the only real reason for showing this slide is that William Herschel was British, so we can claim priority on this part of the electromagnetic spectrum. But in reality, the real work for NIR and agricultural took place in the 1980s by Dr. Carl Norris, who was really the hardware guru there, and developed a lot of the hardware from which our current technology builds on, and Dr. John Schenck, who was the chemotrician and has provided many of the modeling tools that we still use today. And really the technology, going back only to the 1980s, is very new. Compared to some of the wet chemistry techniques such as Keldol, which was developed in the 1800s, we really are at the start of this technology. And just to give a brief introduction to NIR and a bit of understanding, I've come to realise over this last week that my NIR black box technology is nowhere near as complicated as your ruminant black box technology. But I just hopefully give you some idea of how NIR works. And if we take a look at what we currently know, the visible part of the spectrum, things that we recognise every day. So our eye is the detector, and that detects the image. That image is processed by our brain, but it's really our knowledge that helps us determine or make use of what that image is. When we're very young, we'll see it as a furry animal. It doesn't take long for us to realise that it's actually a dog, but it actually takes quite a few years from that stage to realise there's many different species of dog, and that this is an Alsatian. And if we ever get to Kennel Club, we'll really know that this formation of this dog, what makes a good and what makes a poor Alsatian. But if we'd never seen an Alsatian before and we didn't know what an Alsatian would, even if we had 20/20 vision, we wouldn't know what that. We could maybe guess it was a wolf, but we wouldn't actually know exactly what it was. So it's really the knowledge that is the important part of this picture, not the detector. And as I said, our infrared comes just at the end of the visible. Typically the near infrared range runs from 800 nanometers to 2500 nanometers, and then moves into the mid-IR, far IR, and then microwave technology. So it's a relatively weak energy form. It doesn't penetrate very far into a sample. It's that sort of warm glow you get from an electric bar heater. But if our eyes could detect NIR, we would see a world of vibrating molecules. And that energy is just at the right frequency to cause molecular bonds to vibrate. And those bonds tend to be atoms connected by a weak dipole moment. So we're talking about organic molecules, carbon, nitrogen, oxygen, hydrogen, where they're bonded together by a covalent bond, and that energy will make a vibration like a spring. And those vibrations take place in a different part of the spectrum, and we can turn that vibration into absorbed energy, and then finally into a reflectance spectrum. And this is typically what an NIR spectrum looks like. And we can see there's definite regions where we'll see absorbances. So in this region here, we're seeing OH bonds. So this is where typically you're going to measure water. In this region, we're seeing carbon, nitrogen, hydrogen bonding. So that's where we would look to measure protein, the COH bonds, and the COOH bonds of oils and lipids. So this is obviously where we would measure fat. And it's not a very nice spectrum from a spectroscopist's point of view, because it's very broad overlapping peaks. We get lots of information overlapping. But equally, it's quite nice in as much as it's an naturally attenuating spectrum. So if we meet saturation point at this region, we can see the same vibrations, nitrogen and hydrogen, occur down here. So we can make measurements in these regions if we're meeting saturation in this. So it has its benefits. It doesn't follow the Beer-Lambert law. We can't actually take the height of the spectrum because there's a lot of co-eluting peaks there. But we can use it for modelling, and it contains lots of information about what we want to see. Energy, protein and water. But it also contains a lot of information about the life cycle of whatever we're measuring. So if it's a plant, we can look at the type of soil it was grown in. We have that measurement in there. We can look at the fertilizers used. That's still contained within that spectrum. The geography of where it was grown, the weather conditions, the harvest conditions, the type of harvest, and the variety. And all that information, along with those proteins, moisture and oil, is contained in that sample. In fact, we did some work for the Primark part of our business, where they wanted to identify the country of origin for the cotton. And we could use NIR to identify specifically where the cotton had been grown. It would work on ginned cotton. Unfortunately, when it got further up the process and it was washed and dyed and woven into a t-shirt, we'd lost that traceability. But we could provide traceability of ginned cotton and identify where the cotton had been grown. So in our schematic, we could replace the image with a sample of corn. Here, the detector is one of our NIR instruments. The processor is a PC. But again, the important part is the calibration data. For us, our brand name is Ingot. So that just means it's the calibration data. And we can then turn that information into a set of results. So we use our calibration, our modeling technique, to change that into a set of generated reports and results. But again, the important part is really the calibration data. How well we've defined all the different types of corn we can see. How many harvests do we have? How many geographies have we collected that information for? Again, we can have the best instrument in the world, but if we haven't got that in our database, we're not going to make a good assessment of the quality. And then in terms of how do we go about making a calibration, what do we need? And we really need to start with collecting samples. And the minimum number of samples that we would even ever consider making a calibration on is 100 samples. But again, we don't want just the first 100 samples that come into our laboratory. We want to try and cover as wide a geography as we can in those samples. If we can cover seasons, and as a rule of thumb, you really need 4 seasons to accurately predict the unknown. So really we want to collect those samples over 4 harvests. It's difficult. So we scan all those samples on the near infrared, and we send the same samples away for the wet chemistry. And depending on what we want to analyze, obviously that can become quite expensive. We bring those 2 bits of data together, and we use the chemometric software, the calibration software, to model the response in the NIR spectra to those different nutrients. So we're producing a prediction. The technique is a secondary technique, and really, again, we need to be constantly maintaining that data. So once we've got this 100 samples and perform the calibration, we can put it into routine measurement, but really we need a process of feeding back more data into this calibration and going around this cycle of updating this calibration on an annual basis and finding samples that don't fit our calibration. And there's lots of techniques we have in our chemometrics tool that tell us whether or not the sample has been seen by our calibration or not. So the NIR not only will tell us the result, but it will give a confidence level on how well it can predict that result. And those samples with poor confidence are the samples we need to go into the calibration and update it. But once we've done all that work, we have huge amounts of benefits from NIR. It's very fast, it's accurate, it's easy to use, it's non-destructive. We can actually use it on whole material, whole grain. Plant breeders use it a lot, where they can actually analyze the grain, select what they want, and then carry on and grow that grain into the next generation. We can look at chemical and physical properties. So we're not only restricted to the chemical compounds, protein and moisture, but we can also look at physical properties such as rheological values in wheat, so extensibility, elasticity in doughs and whatnot. Identification analysis, and also authenticity, country of origin, where it's grown from, is it contaminated. So there's lots of advantages to us sticking with NIR. It's a great analytical tool, covers lots and lots of different functions. At AB Vista, we work with all the different instrumentation. We work with every commercial maker model, be it a benchtop NIR. This one here is a online system. So the NIR is actually placed in the process and taking constant measurements in feed mills and flour mills, and also the portable type of equipment that I'm going to talk about. So what is NIR for, our near infrared? You've seen it over there, it's on display there. As I said earlier, it's based on a linear filter. It's a wedge, it's a prism, so we create a spectrum of light by shining light across this prism and collecting all the different wavelengths. Our instrument works from 920 nanometers to 1750. So it's this white line. The blue line is what typically is the spectrum from a lab-based, bench-based instrument that runs from 1100 to 2500 nanometers. But as I said before, there's a lot of repeating spectra. We specifically selected this region of the spectrum for our work on forages because this higher region, this is a very large water peak, this gets saturated very quickly. Water has a big impact in the NIR region. It is a very strong absorber. We get a lot of information from water. In fact, there's a whole science called aquaphotomics that just looks at the water part of the NIR spectrum because it can magnify certain compounds within the sample. And in fact, we've done some work with Edinburgh Zoo where we've looked at panda urine to identify the estrogen hormones and identify if the panda's pregnant just by looking at the water peaks in the NIR region. But I said this is exactly the region we're working in, and we've got calibrations for silage, grass silage, corn silage, whole crop, which is whole wheat and siled, and also fresh grass. So we're looking at the whole range of forages. We also can work with feed and ingredients, but specifically today I'm talking about the forage. So I want to talk really about the analysis and some of the pitfalls that we do see with forage analysis. Specifically, there's a lot of debate over whether we go for dry forage analysis or wet. In both cases, we have to take a sample. On dry side, we dry it overnight at a very low temperature in 4-strath oven and record the dry matter loss. We grind it, scan and analyze and report. And typically, dried forage analysis by NIR will take 24 hours. There's a huge benefit if we can do it wet, if we can prove we can do it wet, because simply we take a sample, we scan, analyze, and we report. So within 1 hour, we've got the full range of analysis on the wet sample. The pitfalls, variability is a big problem, and this is variability whether you're looking at wet chemistry, dry NIR, or wet NIR. And this is work done by Cornell Nutrition, and they took different samples in this W shape across the face of a silage pit or silage clamp. And when they analysed those individual samples, they found obviously a range of dry matters. And you can see we have a wet area on the top of the silage pit. we have this drier area in the middle, and then obviously this very wet area at the bottom of the pit where all the effluent gets washed down to the bottom. But the effect of that is a major issue. So if we look at the difference in dry matter content between these 2 points, going from 35 to 25% dry matter, that equates to 7 litres of milk per cow. Even a relatively small difference, 33 to 36%, is the equivalent of 2 litres of milk per cow. Sorry, I'm working in litres, I hope you forgive me. Energy, we work on metabolizable energy, and again, a small change in energy between 2 points is the equivalent of 2 litres. And a lot of what we've been talking about today, the difference in NDF between 2 points on the same clamp can give us serious health problems. So that variability is huge, even across just that simple face. And as you go back into that pit of silage, that variability will get even worse, even higher. And Trowell reckoned that if they didn't account for any of those changes in the season, that would be up to 500 litres of milk per cow per winter. And that's a huge amount in a typical herd. So we really have to look at how we can deal with that variability. And this is just variability in sampling. There's nothing to do with the analysis yet. And there's a little simple statistic that tells us how good we are at defining the average value of that clamp. How good can we get an average value of dry matter for that clamp? And if we look at that model and look at the dry matters and take the standard deviation of those dry matters as the standard deviation of the population and put it into that equation, if we were just to take one sample of silage from that clamp and analyse it for dry matter, what we would have is a result that is in plus or minus 8.2% of the true mean of that silage clamp. If we took up to 9 samples, which is that full W, then we do get a sensible mean. We'll be within 2.7 of the average, so it would be an average plus or minus 2.7. So when people take 1 or 2 samples from a clamp and send it to different laboratories, it's not surprising that we can come up with quite different results on what is essentially or typically what people think is the same sample. But of course, it isn't the same sample. We can see that variability across the clamp, they are very different samples. And similarly, when we get the sample into the laboratory, we have to do another subsampling. And in this experiment, we took a 10 kilogram sample and split it into 10 1 kilogram samples. Each sample we analysed once on our lab-based NIR, and the range between the highest and lowest recorded result for that 10 kilogram sample, if we did single analysis on each 10 samples, was 6.6% dry matter. If we did that in duplicate, so we took the sample, each 1 kilogram sample, and analysed it in duplicate, the range across the 10 samples we could reduce significantly down to 2.8, and if we went up to 5 samples we could significantly reduce the range. And that was just not on dry matter, but that was on all the different nutrients. So we can't just, when we get the sample into the laboratory, we can't just take one sample. We need to take a whole, at least duplicate, if not triplicate, would give us the best set of results. But of course, in a laboratory environment, that's not easy, especially if we're doing wet chemistry, because that's very expensive and very time consuming. And just to show it wasn't a fluke, we did that on a number of different samples, and again we see the same thing. The more times we analyse duplicate samples, the better we are at getting to a good average. This is just to show our NIR result for, this is dry matter, sample A. I've got total dry matter, sample B, sample C, so you can see where the NIR fits against other laboratories doing the same thing. This is a fairly horrible slide. In the UK, we have a forage advisory group. So all laboratories in the UK undertaking forage analysis use exactly the same calibration. We have a national calibration for forage. It's just one calibration, or it's a calibration for grass silage, a calibration for maize. But every test in the laboratory uses the same sample. Every month, we send out 10 samples to each of the participating laboratories, and they report back their results. So this was just looking at variability. We range across laboratories. If we go down laboratories, the agreement between the labs, the correlation between labs is very good. But across laboratories on any one sample, the range was going anything from sort of 3% NIR to almost 10% NIR. So a laboratory using the same calibration on the same sample was reporting results at least 10% different. So we tried to do some work with these laboratories to try and improve that discrepancy and reduce that range across the group. So we've done a lot of work on standardizing the instrument, standardizing the technique. But we came up with a technique called repeatability. So in this case, what we do is scan the same sample under different conditions. Here we use the different instruments as the conditions. So we're saying scan the same sample on many different instruments. It could be temperature. We could scan the same sample at different temperatures if we wanted to eliminate the effect of temperature in our calibration, or technicians, or any number of things. But the idea is that you have exactly the same sample. So these were sealed samples in sealed cells that you could take and scan under different conditions. And then you put those samples into the model. And your modeling techniques then allows you to make the best model with the highest correlation, but the lowest effect on that parameter in your repeatability. So we let the model choose what was best in terms of accuracy, but also in terms of precision across multiple instruments. And fortunately, in every case, we could significantly reduce. So the light blue is the calibration before repeatability file, and the dark blue is the error. I mean, there's some big differences in grams per kilogram. This is in percent. I should have used different scales. But in every case, you can see the dark blue is lower than the light blue. So we had a modeling technique that we could also use to give us better precision across different instruments. And we use the same technique with the NIR4. And again, here we can see 4 different instruments. This is the serial number of the different instruments and how we can improve the repeatability of those instruments. They actually come out of the factory very well matched. The linear filters are very closely matched. So in terms of this piece of work, we're starting off with a very variable material, and really the best way of dealing with that is to take multiple samples. It's costly, it's expensive, but it will in the end pay dividends on what we want to do. So our primary sample needs to be made up of many different samples. In the laboratory, we need to do at least duplicate analysis. Triplicate would be best. And then if we've got a network of instruments, we have techniques that we can use to help us across that network of instruments. And now we've got 200 NIR instruments, NIR4 instruments out in the field. We have a very good sense of how well we can replicate those calibrations across all those 200 instruments. The message is really that there's a lot of noise in the system. If we look at comparing one sample analysed in 2 different laboratories, we'll get differences. We can get some quite big differences, as you can see. But really, we need to get through that, and we need to really try to be looking at these step changes rather than just the noise in the system. And the clear way of doing that is to take more samples, analyse more data, and we'll get a better idea of where those step changes really are. This is just to give you an example of the dry matter calibrations for the grass, maize or corn silage and fresh grass, and the type of errors that we're seeing. So typically plus or minus 1.8 between the NIR and the wet chemistry, and RPD of 8. So we're looking, we want these RPD values to be greater than 2 really to be a very good, robust calibration. So in every case we've got an RPD greater than 2. And these areas are what we would generally see between 2 laboratories undertaking the same analysis on the same sample. As I said, this is what the kit looks like. It comes in a case. In the UK, we've got a whole range of parameters. We're working on those now for US forages. We have a very robust calibration for dry matter. We're very happy with the dry matter calibrations, but if I go back to those original slides, we really need to get that information into our calibration set so we're sure we're giving good values on the other parameters, and that's currently what we're working with Dairy 1 on, to improve the range of parameters that we have capabilities for. All that data is connected to the web. As I said, the instrument is totally standalone. You don't have to connect to the web, but if you want to update the data, get the benefit of new calibrations, then that synchronization with the web is there. And then this NIR is just one of many, many different sensors that we have available to us today. We've now got this data based on the cloud. I think the next real step is to be able to merge all that data from all the different sensors, the ones giving us milk yield, the ones giving us information directly in front of the cow, and have some sort of machine learning algorithm, some sort of data mining that can bring all that different information together from those different sensors and help us make better models, not just calibration models, but better models about how to feed the rumen. So as I said, you know, we really need to stop worrying about the noise and really move forward and look at what we're really wanting to measure. And that is, we need to take more measurements, make it easier and simpler. The value to the dairy farmer, obviously if we can better predict the value of the forages in front of them, we get better information, better informed of the forages being fed. And there's a lot of work being done on mass balancing, so being able to look at those. We've got a technique that we can use on the forages, but we can also use the same technique on measuring the manure, and so we can get this nitrogen balancing. There's a lot of information, a lot more we can do with the NIR. With that, thank you.
Thank you, Chris. Any questions for Chris?
Can you transfer the result to the NIR ration formulation software?
Yeah, on the web portal, there's a function to transfer the data into an Excel or CSV format that you can then pull directly into your ration formulation system.
Any more questions? Chris, I've got a question now. In this symposium, I think, has been a lot of discussions about particle size. How far are we from measuring particle size as well, or getting at least a distribution of a sense, you know, to be able to address that?
It's a good question, because particle size is one of the big factors influencing NIR. So if we have a range of different particle size, we'll see a shift in the spectrum. We're not actually measuring a compound, but because particle size will scatter light in our near-infrared spectrum, we do get a shift. So we've done a lot of work looking at corn. So in feed mills, at the gristing of the corn stage, we can actually measure the different particle sizes of the gristed corn. And we have that on the NIR4 as well, another part of the calibrations we have.
Okay, excellent. So how about ruminant and doing that?
We haven't done that work yet. We've done some work on density, being able to measure density at the face of the pit. But we haven't done any work on fibre length. I'm sure it would have the same effect as the particle size. We'd see a shift in the spectrum, so I would imagine we could do it. We just haven't got that data or looked at that bit of work yet.